It's ongoing. It started.
Good morning, everybody, and welcome to the fourth installment of the CASTIEL series Code of the Month. Today we have here Arnaud Montagu. Arnaud is a biologist by education, but he also holds a PhD in applied mathematics, which is a very rare combination. And today Arnaud is working at the computational biology group at the Barcelona Supercomputing Center. And he will present us the code PhysiCellX, which is developed in the COE PerMedCoE for modeling of 3D tissue and maybe up to the organ level in a scalable way. And it's used for computational biology problems and also personalized medicine. And now we are very, I'm happy to hear this very interesting talk. So Arnaud, the floor is yours.
Yes, thank you, Gundram, for the very nice introduction. So let me share screen. Hopefully, yes, you should all be seeing a slide now. So thank you for having me from Barcelona Supercomputing Center and for having us from PerMedCoE presenting this code to your community. I will talk briefly about what PhysiCellX and how we have expanded it in order to be HPC ready. This tool is a multi-scale simulation tool, and we have been using it in personalized medicine use cases. So, So PerMedCoE is a center of excellence that lies in between two other centers of excellence in Europe. So a center of excellence is namely a way to accessing or an open door from the HPC to given communities. So when we started PerMedCoE, there was already CombioMed, center of excellence that focuses more on, let's call them physiological level models, such as fluid dynamics, like the one you can see here of a heart simulation from a team from BSc. And there's also a BioExcel center of excellence in which they focus on molecular dynamics in order to have atomic level models. That is the ones that you can see here, so protein docking, etc. So we decided to focus PerMedCoE in between the physiological level and the atomic level, so what we call the cell level models. And this is a stack of models that go from signaling pathways to metabolic pathways to cell-cell interactions and also cell environment interactions. So when you decide to model in biology, there are two variables that you need to decide before heading towards modeling. One of these variables is what level you want to model, right? I mean, from the very tiny DNA up to cell populations, up to organs or tissues. This is let's say one decision that you take. Depending on the decision, you would take one modeling framework or another, or one tools or another, etc. And then also you need to think about the who is going to use this simulation now, or in what architecture you want the simulation to run. So in computational biology, we have extensively been using workstations, and we have started since many years ago, we started using also HPC facilities. And depending on where the tool is well suited to, I mean, you would tend to use one tool or another. I mean, there are many tools that work extremely well in workstations that don't scale at all in HPC. And of course, HPC ready tools usually have a huge overhead or a big overhead when working on workstation. I mean, it may not be the case for your specific tools, but some of these you need to think about who is going to use them in order to decide what tool you're going to use. And also, in terms of once you're building the tool, you need to think on what user interface you need to have if you're going to promote your tool for workstations or for HPC facilities. So there has been and there is an interest into this kind of simulations or simulations in computational biology, namely because it is thought as a proxy in order to have animal-free preclinical trials. So the FDA recently allowed and even promoted to reduce the animals used in these preclinical trial tests. And the EU is expected to follow or is discussing when to follow or when to allow for this. So the idea here is that we scale up from the organoid simulation to the tissue simulations and in the future to organs organ simulations, with the aim of helping these kind of preclinical tests, and in the very, very long term to have full body digital twins of humans. So of course, in order to transition these different scales of number of cells and complexity, is not just the number, but it's also that bigger amount of cells usually have correlations that are big more complex than one cell interacting with another. In order to reach all of this, you usually need to scale up to high performance computing architectures. And namely, I mean, of course, if you want to think about real size tumor simulations with immune cells interacting with clonalities, with different kinds of copy number alterations, etc., for this you will need bigger architectures than your workstations. So we covered this in a review that we published a couple of years ago, in the sense of what do we have on the field in order to go towards that goal, and what do we need from the field in order to reach it. So PerMedCoE humbly tries to cover some of these distance by optimizing 4 different key tools. So you have them in here. It's PhysiCell, it's Cobrexa, Selenop, and Mabos. PhysiCell aims at agent-based modeling framework, so multi-skill simulation, let's say, from cell populations to tissue, let's say. Cobrexa aims to model cellular metabolism at genome scale, so it's an exascale ready tool to simulate flux balance analysis and flux variability analysis, etc. And then we have 2 different tools, Selenopt and Mabos, that target signaling pathways. So Selenopt is more on the signal transduction networks, and Mabos is focused on the stochastic simulation of Boolean models. So one of the pillars of PerMedCoE was to optimize these 4 different tools to HPC and exascale with the help of HPC experts, and also help from other centers of excellence that aim at the optimization of different tools. So we followed HPC and exascale guidelines in order to adapt these different tools into exascale architectures or pre-exascale architectures. Also, the second pillar for PerMedCoE was the use of building blocks and workflows, in the sense that we know that those 4 tools are not the only tools that could benefit from this exascale. So, and we know that in order to solve interesting use cases, they need to be coupled with different tools that, let's say, digest input data, and also maybe after the simulations, they provide some interesting figures or interesting pipelines. So in order to have this set up, we containerize the different tools, and we combine them with the different tools from the field that are not our own, that could be also containerized, and devise different pipelines and workflows in order to solve different use cases that are of interest to us. So these use cases would be like a third pillar of PerMedCoE, in which we have from the omics-based analysis of cancer diagnosis, to drug synergy studies, to tumor evolution based on single cell, to, of course, there was a COVID multi-scale modeling. I say of course because this project started just before the COVID pandemic. So of course we consider that it was a nice opportunity to have one use case more oriented to COVID infection. So these different use cases are solved by some of these workflows. In fact, from one use case we have different workflows, but we provide these different building blocks and workflows to the community so that they can mix max, and they can plug and play whatever building blocks they want in order to solve their own use case. And this is all open source. This is all containerized in let's say open source standards. We do have different workflow managers that coordinate everything under the hood, and we have an orchestrator that allows you to send part of the pipeline to some cluster, and another pipeline to other clusters. So focusing on PhysiCell and multi-scale modeling, what you need to know is that we consider cells as agents. So here what we follow is the center of a sphere, and this is an abstraction. We know that a cell is not a perfectly round sphere, but is an abstraction that helps us build on top of it in order to solve interesting questions. So these simulations have different agents with different cell properties, and we also have a specific delimitation or description of the environment, in which we can have environment in 2D and 3Ds, and this environment can be perturbed in different several ways. So we took this PhysiCell code from Indiana University collaborators, and what we did was to integrate in each one of the agents to integrate a Boolean model of this of a signaling pathway. So now we can add another let's say time scale in which now we can regulate for the signaling pathways. And these simulators are called multi-scale because you work with different time scales of activities, let's say of physical behaviors. One is the diffusion, another is of cell movement, another is of cell processes, namely how the cell growth when going through the cell cycle, and we have another for signaling. So whatever is happening outside of the cell can affect some proteins that are inside of the cell, and that they activate or deactivate others, and then this leads to several phenotypes at the end of the signaling process. So at each time point, you will query one or several of these solvers, and here you have the simulation main loop, let's say pseudocode, in which you have an update of diffusion that happens many more times before the cell movement. Then once you have 60 times the cell movement, then you query for the cell processes, and so on and so forth. So when thinking about the, as we will see in a couple of slides, when thinking about the use of time of each one of these solvers, you will see that there are several solvers that are queried many more times than others. So integrating these stochastic Boolean models allows us to personalize models according to patients' data. So this means that according to the mutants or the expression of different values from patients, you can let's say tilt the models towards a given phenotype, or you can assign mutations to the nodes, so that at the end what you have is a set of patient-specific logical models that are more or less similar among them, but they have quite specificities, different specificities depending on the omics data that you have used. So for instance, here on the left, what you have is not patients, it's cell lines data, but the rationale is the same, in which you have different cell lines that behave differently according to their specificalities in mutations, RNA, and random initial conditions. So that is one thing that we can work on, and the other is the combination of drugs. So now you can perturb these intracellular signaling pathways, and you can do combinations of perturbations, and you can work with dosages of drugs, and you can combine those dosages of drugs so that you can study synergies of drugs. So you can see that, I don't know, IC70 of this drug combines very well with IC50, and you have a huge synergies in this drug. In this case, it's not 50 and 70, it's more 90 and 50. That is already quite synergistic of these lumicelib and pictivicib, which is prostate cancer drugs. And yeah, so in the case that you want more information on this, I have just let here the references of this. And also, interestingly, PhysiCell allows to study complex environments and also, I would say, dynamical drug regimes. So in this paper here, that very recent, I mean, like 3 weeks ago or 2 weeks ago, what we did was perturb or have a dynamic drug regime in which we change the frequency of adding a given drug that perturb the signaling pathways. And what you see is that there are some frequencies that are better than others if we want to increase the necrosis cells, which is this brown line that you see here. And in green, what you have is the alive cells. So when these frequencies is very far apart, you see that the cells keep growing. While if you have like smaller frequencies, you will see that the necrosis takes over the proliferation of cells. And also we can combine extracellular matrix definition in order to study the different migration patterns of the cells. And we can see that according to the different densities of extracellular matrix and properties of the cell, you can have different migration modes colonizing, for instance, breast cancer. So this was more, let's say, on the biological side, or on the biological motivation and the usefulness of this simulation. But let's move now to the more computational science side of the project. Here I will be showing this kind of figures, which are traces, which is that we have extracted using 2 different tools that have been developed at BSc that are called Extrai and Paraver. And here what you see is on the x-axis you see time, on the y-axis you see the different threads of a computing node or several computing nodes, and different colors, you see how much time the runtime is in given some given functions. Okay, so you have, for instance, here in pink, you have diffusion. Here on red, you have mechanics. Here on green, you have computing the gradients of the diffusion, and so on and so forth. And whenever you see black lines, this means idle time. Okay, so what you can see, this is a part, this is a trace of PhysiCell in which you can trace back, let's say, the different pseudocode into these traces. So you first have diffusion many times, and you have one time of mechanics, and you have another diffusion many times, and you have one's mechanics, etc. So in fact, what you are is in these times. You're not even reached the update cell processes time. And what the first thing that we did was taking just one computing node in Mare Nostrum. We increased the number of threads, and we studied the same focus of activity. Let's say one diffusion time with one mechanics. And what you can see is that when you are in low numbers of threads, the diffusion takes most of the time. But then once you increase the number of threads, this pink part is just reduced to 10% or 7% of what it used to be. But interestingly, what you see is that the mechanics part is being increased, which doesn't make much sense. I mean, in the sense that you would expect that you have many more workers, so that the runtime of this should be compressed, as you see in the peak. And in fact, what happened here is that we have a bottleneck in the memory allocation, or in the reading and allocating memory. Why? Because the way that the vector of cells is written, whenever a cell divides, you add the daughter cells at the very end of the vector. So in these mechanics, what you're looking for is that you're integrating your movement with the neighboring cells in order to know where you're going to collide and where you're going to go after that. So that means that after some time in the simulation, your neighboring cells are all your daughter cells. So that in order to reach for this neighborhood, you need to read a lot of times this vector up and down. So that was one of the drains of the cause of the drains of these traces. And in fact, this we corrected by just one line of code, or 2 lines of code, which is the memory allocation library that we were using. So let's say in the initial setup, or the default setup, we were using malloc library, and just by changing it to malloc library, we increased 6 times the scalability of this tool. So here on the y-axis you have speed up, so normalized for 2 processes as baseline, and on the x-axis you have number of processes. So the ideal line is that whenever you double the processes allocated to a task, you will halve the time of this task, which, as you see, did not happen. And here we have increased the speed up by 6-fold, but still we're a bit far out from the ideal. I mean, it's very rare that you tackle the ideal unless the code is highly optimized. So this was done by Marc Klaska and Marta Garcia Gasulla from the POP Center of Excellence, and this allowed us to publish these findings in a conference, in a programming conference. And what we did here in the paper is to propose several ways of increasing the scalability of this tool. So in this case, what we did is we have different versions of the code in which we have added one more change. For instance, the malloc library that I was talking about, but also the collapse, adding collapse clauses in nested loop, or leaving space in between cell vectors. And you see that some bring you closer to the black ideal line. So for instance, the yellow one is the one that is closer, but once you add the parallel voxels to the yellow one, you see that the blue line is slightly underneath the yellow line. That means that not all the changes that you do are increasingly bringing you towards the ideal. Sometimes you screw up your runtime. But interestingly, what we saw, I mean, this you can imagine the way that these simulations work. They are memory bound. I mean, there is a point where you are allocating cells to your memory, and then at some point you just run out of memory in the node. So this is for one node in Mare Nostrum 4, and after several hours, you see that the simulation is just stopped because there is no allocation memory possible. So this led us to think of how we can distribute these simulations among several computation nodes. And for this, we implemented message passing interface into this code that already had this OpenMP standard. So here the idea is just, I mean, if you slice a bread, I mean, we're slicing the simulation domain and we're allocating each one of these bread slices to a given node. And in between, to move from the bread slices, you use message passing interface. So there will be information that will travel from one computation node to another. And whenever you're simulating into one of the subdomains, you're in one computation node. We decided to divide the x-axis. So whenever you travel through the x-axis, you're traveling through computation nodes. And whenever you're traveling through y and z, you're staying in the same subdomain. So in this case, whenever, I mean, it's very fast to do things in the x and y plane, but whenever you're moving to the x, it's a bit more costly. I mean, you do have this overhead of the NPI. So we wanted to study this overhead, and in fact, we were a bit surprised to see that that it was still efficient. I mean, when you compare the hybrid, so the OpenMP plus MPI version with the pure MPI, you would still see similar times. In fact, in some processes, the hybrid was a bit faster than the OpenMP. But the reasoning or the goal of doing this implementation was not to compare it to the one node, to the serial, let's say, code, but to enable us to have huge simulations or bigger simulations, which is what you see in this table. I mean, this domain could not be allocated into one node, and we work on the hybrid implementation in order to study bigger, more complex simulations or bigger setups. Still, of course, I mean, as you can see, the simulations are memory bound. I mean, we do have issues in terms of memory allocation, but now we do also have other problems of optimization of these hybrid simulations. So in fact, these are two very simple simulations. On the left, you have a two-dimensional simulation of predator hunting prey. So it's kind of like a Lotka Volterra simulation in which the predators are being hunted by different prey. So in red, you see the predator. In red, you see the prey, and in blue, you see the predators that are hunting the prey. So this is a bit unfair because here we compare one node against 10 nodes, and we see that, of course, with a huge amount of nodes, I mean, with bigger amount of nodes, we cut down the efficiency. I mean, we're quite efficient. The speed up is fourfold, but the efficiency is not very good. It's only 40%. And in this case, we have a 3D simulation. Imagine a shoebox that is full of cells just dividing, and nothing is impeding the cells divide. And this shoebox is already 50 times 10 times 10 millimeters size box. And this is also a simulation using 10 nodes. And so these simulations, this tool, we have tested it in Mare Nostrum 4, also in a Huawei cluster called Kupeng that we have also at BSc, and also in several other pre-exascale clusters that we have in Europe, such as LumiC, Hawk, and Marconi 10. But going back to this efficiency, we were a bit worried about this efficiency. I mean, something was happening here that was draining the runtime of the simulation. And for this, two engineers in our group, Jose Estragues and Thalia Diniaco, started working into finding this memory drain. And at the end, what we find out is that whenever we were using the diffusion solver, which is the one that you call the most often, this diffusion solver solves a tridiagonal matrix in which the X needs, I mean, you do a forward substitution, and then when you finish the forward substitution, you could do a backward substitution. And at any time, X needs the values of Y and Z, which is why you do the backward substitution in order to finish its run. So whenever you are in shared memory, these dimensions X, Y, and Z are quite, I mean, they are quite compact, let's say. But once we subdivided this among X, I mean, imagine that we were traveling here among the bread slices. You see here very clearly the different bread slices of the forward substitution and of the backward substitution. So the second subdomain in X cannot start until the first subdomain has finished. So this basically serializes the X dimension in this setup, which is not ideal. I mean, in fact, it's quite bad for the runtime, because this, when you increase subdomain, this increases the problem. I mean, there are many more nodes that are idle waiting for the previous node to finish. So what Jose did was to overlap communication and computation, in a sense that you divide this subdomain into blocks, and then you run one block and you send that block to the following subdomain, so that the first block of the second subdomain can start, and so on and so forth. So this cuts down this V shape to something that combines X and communication and computation of X, and then the Y can start before, and of course the Y in comparison with the previous one ends after, because it still needs something from the other blocks. But in global, we go down from 2.6 seconds for this amount of work to 300 milliseconds for exactly the same amount of work by overlapping computation and communication. And not only that, but in order to facilitate this computation and communication, José Estragues, what he did was to change the data structures of the diffusion. And these data structures allow us to allocate bigger simulations in the same amount of nodes in comparison to the previous version. So here on the left, you have a number of substrates. On the X, you have size of simulation. On the Y, you have number of substrates. And in color, you have number of nodes. So let's say the PhysiCell that is not distributed is just the color of one node. So you can allocate up to 2 substrates in a 216 voxel size cube. And this is the most that you can do. Once you work with distributed versions, you can go up to 96. In this case, we have tested 96 nodes. And here you have the number of substrates, and then you can use many more substrates in many more nodes, in many more voxels, but still you are constrained. I mean, there is a part of the simulation that you cannot allocate. And by having this overlap, what we see is a much clearer diagonal in which we can now allocate many more substrates in many more sizes, let's say, of simulations. But still we need to use even more nodes in order to allocate this 800 voxel cube. So yeah, so this was, I mean, just to tell you that the simulation is still memory limited, but there are ways of, just by changing your data structures, to have increased performance in this sense. So the last thing that we did was to test the diffusion, to do some scalability test of the diffusion part of PhysiCellX. And for this, what we did was to devise different workloads, and then workloads that the bigger one is already 6 times smaller than a whole liver. So we are already there in the, let's say, in the meso part of the organs. And then we increased the number of nodes, and we tested the wall clock time of these workloads in this number of nodes. So you can imagine, for small workloads, smallish number of nodes is better than bigger. I mean, there is a point where you don't reduce the runtime by having more nodes. I mean, the overhead just kills you. And then as you are increasing the workload, you see that this number of nodes that is ideal is increased. And at the end, this massive workload that has a minimal runtime in 64 nodes. So in order to be useful to use this simulation in 96 nodes, we would need to have bigger simulations, not only of size but also of substrate. So the number of substrate also multiplies the number of diffusion that you need to do. So, and this was done using these overlaps. So we're comparing in green, we're comparing the one node, let's say, simulation, and in orange, or in orange, what we have is the diffusion part with the communication and simulation overlap. So the perspectives of this work are to keep doing extensive tests of this code in bigger scalability in other architectures, and also looking at the energy of this simulation. This is one part that we are quite interested right now. And then, of course, we are combining, or we're trying to combine some parts of the code with different accelerators. So we do have an alpha version of a GPU that is working on these kind of problems. And we're also looking how we can vectorize this code, and how we can use, or if it would be interesting to use tensor computing for this. But this would also need a major refactoring of several parts of the code. So yeah, so we're thinking of different code design strategies for this, namely with FPGAs and also with different architectures. And we're very open to learn from any success cases that there is in the field. And lately, what we have been working is to collaborate with developers at BSc that they have a tool called ALIA that simulates organ level. They do organ level simulations in which nest our simulations underneath their simulations in order to simulate whole organs from the proteins to the organ. Let's say that you have different levels of perturbations that you can change and you can study. And interestingly, also, we have teamed up with different developers in the field in order to benchmark our multi-scale tools, to set up different unit tests and test cases, and to have some performance tests of these tools. So here, the goal is not only performance, but it also to be sure that we're simulating the same in the same way, which is something that is still a lesson that we need to take in the field. So that's it from my side. I mean, of course, I have been presenting this work, but this has been a teamwork from people from BSc and from people from QE, and also thanks to our collaborators in order to provide us with tools, with data, etc. In case you have any question or any comment, please you can contact me in mail or through Twitter, also known as X. And yeah, that would be all from my side.
Thank you. Thank you very much, Arno, for this very interesting talk where you have really nicely presented the challenges also and the potential of this technology. And the floor is open for questions. So you can, as I said, either type them in the chat or use a specific question tool.